All right. Good morning, everyone. Thank you for joining us for Home Care Assistance's Dementia360 speaker series featuring Dr. Howard Fillit and thought leaders reporting from the front lines of dementia. Alzheimer's disease and other types of dementia touch hundreds of millions of lives, from the people living with the condition and their loved ones to professional caregivers and healthcare teams. Our goal over the next 2 weeks is to provide you with a comprehensive perspective on the most up-to-date research, caregiver support, prevention tips, and more. So no matter who is impacted by dementia, you will walk away with tools that will be helpful to you or someone you're caring for. I'd like to turn it over to Dr. Howard Fillit. Thank you so much for joining us this morning, and I'll let you take it from here.
Okay, thank you very much, and thanks for inviting me today. I'm Howard Fillit. I'm the founding executive director and chief science officer for the Alzheimer's Drug Discovery Foundation, and I'm also a clinical professor of geriatric medicine and neuroscience at the Icahn School of Medicine here in New York. And I've spent most of the last 40 years taking care of people with Alzheimer's disease as a geriatrician and doing research and developing new drugs for the disease. And just to give you an overview, I've never been more excited about our field than I am today, and I hope to give you some sense of that optimism during the course of my talk. If I could have the next slide, please. As was just mentioned, we're facing a huge epidemic, next slide, of this neurodegenerative illness. Right now, there's about almost 6 million people in the United States that are suffering clinical symptoms with the disease: dementia, mild cognitive impairment, which is a significant number. It's become the 5th leading cause of death in the elderly over the last couple of decades. Interesting recent study estimated that there could be as many as 47 million people that have Alzheimer's disease in their brain, the plaques, the typical plaques and tangles and other pathologies, without actually having symptoms yet. And so what this means is that the number of cases of clinical symptoms with dementia could grow to almost tripling by the next few decades. So this is a disease which is one of the only chronic diseases of old age without effective therapies. It's a chronic, progressive, uniformly fatal neurodegenerative illness, primarily of the elderly. And so we need to conquer it with new drugs. It's also, in terms of financial and economic burden, the most expensive disease in developed societies throughout the world, primarily because of the burden of long-term care, but also hospitalizations from aspiration pneumonia, bed sores, falls, hip fractures. And this affects individuals, of course, in a huge way, as many of you know, working with home care assistants, of course, how important home care is to the quality of life and safety of all of these people that are having cognitive impairment and losing their minds, but also their loved ones who are caregivers who are undergoing tremendous caregiver burden on a daily basis. So we can't help those people, but we need new drugs. So what do we know about the disease and developing new drugs? And if I could have the next slide. Well, one thing we know for sure, aging is the leading risk factor for Alzheimer's disease. And so what does this mean in terms of our scientific strategy for developing new drugs? How can the aging process and our burgeoning knowledge of the aging process lead to new drugs for Alzheimer's disease? And in the next slide, what you see here is that it's interesting that all of these diseases, heart disease, cancer, stroke, emphysema, pneumonia, and so on, Alzheimer's disease, they're all on a similar trajectory through life. They're all increasing with age. And so the underlying fundamental process of aging itself strikes different organs in different ways, but it's likely that these fundamental processes of aging are what is causing all of these chronic diseases of old age, and Alzheimer's is no different. And the value of this perspective is that it can give us new insights into developing new drugs for Alzheimer's disease in a couple of ways. One is we can learn from, for example, heart disease and cancer where we have effective drugs, and try to reapply those drugs to the treatment of Alzheimer's disease. And this is called repurposing. Repurposing drugs that are already on the market for other chronic degenerative diseases of old age for Alzheimer patients. And I'll get back to that in a moment. but also by understanding the biology of these other diseases, we can develop new drugs for Alzheimer's disease that are specific. So this is really important, and it's been our strategy for 20 years in terms of how to develop new drugs. Next slide, please. Now, these processes, and I won't go too much into this because this starts to get a bit technical, but I'll try to highlight the importance of 7 of these processes. So one of them that's taken on the most interest in pharmaceutical industry over the last 20 years has been what's called here misfolded proteins. And you all know that, or perhaps you don't, but one of the most obvious changes that Alzheimer's himself described in 1906 in the brains of people with Alzheimer's disease were these deposits of plaques and tangles. And these are deposits of what are called misfolded proteins. What that means is that proteins have to be in their normal 3-dimensional structure in order to function. And when they're not, when they're misfolded by various processes like inflammation and oxidation and so on, when these proteins get damaged, they misfold, they become toxic to the neurons in the brain, and they deposit as these plaques and tangles. And these targets of the plaques and tangles for the last 20, 30 years have been the primary focus of drug development in the pharmaceutical industry. And that's how Aduhelm, which is the first drug ever to be approved by the FDA as a disease-modifying drug in history recently, came to market. And we'll talk about Aduhelm a little later. But it's an example of trying to get rid of these toxic misfolded proteins. We know that the aging brain and the brain with Alzheimer's disease has inflammation. And so knocking down that inflammation that can lead to cognitive impairment is really important and is an emerging target in Alzheimer's disease. Epigenetics means that our DNA actually gets, and the process of how that DNA is used to make proteins and make our bodies work, actually gets damaged with aging. The DNA doesn't get mutated necessarily, but it gets damaged. And so the machinery of the DNA, the essential element of our lives, gets damaged. And so we can try to fix those epigenetic changes that happen to the DNA with drugs, and those are in development. Another one I'll talk about briefly here is mitochondrial and metabolic dysfunction. We know the aging brain is affected by insulin resistance, the common denominator of diabetes. We know that diabetes is a risk factor for Alzheimer's disease. We're currently repurposing diabetes drugs for Alzheimer patients to see if by improving the insulin resistance that exists in the aging brain can improve cognition and maintain neuroprotection and neurons. The brain is amazing. It's only 3% of the body weight, and it uses 25% of the body's energy at any time from glucose and oxygen. So even the slightest decrease in the ability of the brain to use glucose through its metabolic mechanisms can lead to neurodegeneration, and this is an important target. Vascular disease, hypertension is a major risk factor for Alzheimer's and other dementias. And some hypertensives, antihypertensives, actually have neuroprotective properties. And we've been repurposing antihypertensives for Alzheimer's in people that don't even have hypertension, and most recently showed some really interesting results with those kinds of drugs. And I could get into that more later, but let's not forget that vascular dementia is the second most common cause of dementia. We're not necessarily talking about big strokes here. We're talking about microvascular disease of the brain that we can pick up on MRI evaluation of patients. And this is an emerging field right now with a lot of interest in new targets. And the synaptic activity one is really the symptomatic agent. So those were the first drugs brought to market, drugs like Aricept or donepezil, Razodine, rivastigmine. These are all drugs that work on neurotransmitters to improve memory function, but they don't change the course of the disease. Next slide, please. How do we develop new drugs? What's going on? Well, I sort of have spoken about that a little bit, but let me just say right now that there's over 121 new drugs in clinical development in the various stages of clinical development, phase 1 being safety in small numbers of people, phase 2 being the first time that drugs are tested for efficacy in patients with Alzheimer's disease, and then phase 3, where it's proven, hopefully, that drugs actually work, and that data can be brought to the FDA for approval. About 80% of drugs that are in development now are in phase 2, which is really exciting because we have a lot of drugs where proof of concept is being conducted. So somewhere between 50 and 80%, depending on how you cut the pie, are in this phase now where all of the basic research that's been going on in Alzheimer's for the last 40 years is finally coming to harvest in the event of new drugs that are being tested in human beings with Alzheimer's disease. And of course, I don't have time now to talk about these drugs, but I will say that there's some really exciting tests coming out, companies that we've invested in that have neuroprotective agents and so on, that could actually be quite revolutionary for the treatment of Alzheimer's and other neurodegenerative disorders. So this is really an exciting time for the field, and a tremendous amount of efforts going in. And there is more money coming into our field now through government and investors and pharmaceutical companies with interest in this, and our own foundation. Next slide, please. This is kind of what our own foundation has been doing over the last 22 years. If you give me a chance to just tell you about our venture philanthropy, we've invested over $168 million in the development of new drugs. We've funded over 650 drug programs throughout the world in over 19 countries. Part of our venture philanthropy model is to invest in startup biotech companies and early stage biotech companies, and even publicly traded biotech companies. And so we've funded 110 programs. We've made over 175 investments in industry programs. And this is very consistent with our model because we're essentially in the developing and focused as the only foundation in the world that I know of that is solely focused. All we do is fund the development of new drugs from the preclinical through the clinical phases. Sorry. That we're unique in being the only foundation. We don't fund education or advocacy or caregiving. The only thing that our group does, we don't fund basic research, is we work with partnering with companies and academic universities to develop new drugs for Alzheimer's. And I think something that I'm very proud of now is that we have 35 active clinical trials going on around the world. About half of these, as I mentioned earlier, are repurposing trials where we're taking existing drugs that are on the market, where we know the safety profile of these drugs, and we have good reason through either epidemiological data or preclinical data in mouse models of Alzheimer's disease or in vitro that these drugs could have effects in Alzheimer's disease. And we then go right to phase 2 and test these drugs that are on the market in Alzheimer patients. And during the last year, we've had about 6 or 7 readouts of these programs, which have been very positive. And our current efforts are directed at taking these 6 or 7 positive programs that we've had of repurposing and help figuring out ways to commercialize them. Many drugs on the market today are derived from repurposing existing drugs. Thalidomide was a good example of a horrible drug in the 1950s for pregnant women, led to all kinds of postpartum problems and fetal abnormalities. But a scientist came along and said, you know, thalidomide has certain properties that might make it good for multiple myeloma. And a biotech company was started called Celgene, which ultimately became one of the most successful biotech companies in the world. And their lead drug was thalidomide repurposed for multiple myeloma. And even today, derivatives of thalidomide are still being used for the treatment of multiple myeloma. And that's just one example. Another simple one is aspirin that was used for pain and fever, and today is often used for the prevention of heart disease. So the other half of our portfolio of drugs are new drugs, new chemical entities that are being targeted at novel targets and are in the usual regulatory stages of development. Next slide, please. Now, one of the really amazing advances that has happened in the last several years has been the development of biomarkers. And biomarkers are critical to drug development. Imagine if we didn't have cholesterol, how would we develop, if we didn't have a blood test for cholesterol, how hard would it be to develop a drug for heart disease? Imagine if we didn't have a blood pressure cuff and blood pressure readings, how would we possibly develop drugs for hypertension? These are biomarkers. They're needed for early diagnosis. They're needed for screening in clinical trials. They're needed as monitoring outcomes in clinical trials to see if the drug is working. And in doing so, if you look at cancer, for example, today, the advances in chronic care management of cancer are almost entirely due to the fact that we have so many biomarkers to phenotype, to type these cancers, which used to be just thought of as tumors, solid tumors on an X-ray in the lung, for example. And today, we take a biopsy of that tissue, we type it with various biomarkers, and we set up a whole bunch of drugs against those biomarkers when they're positive in that patient, and we give multiple therapy for that patient based on their biomarkers. That's called precision medicine with the biomarkers, and combination therapy to attack the multiple pathways that these diseases of aging have that we need to overcome. And that's where we're going with Alzheimer's disease. That's the importance of biomarkers, the critical importance. And there's been a revolution in biomarkers in Alzheimer's disease. So let me go to the next slide. And as I mentioned, biomarkers are not only, as we see here, tests to diagnose the people that have symptoms, but one of the things that biomarkers such as PET amyloid scanning, PET tau scanning have shown us is that by being able to see Alzheimer's disease in patients, in living patients, to see the plaques and tangles with these biomarkers, we've discovered that Alzheimer's actually begins as a disease of middle age, and that the amyloid deposits, the plaques that are typical of Alzheimer's disease begin in people's 40s and 50s. And it's not until they're in their 70s, usually 20, 30 years later, that they develop symptoms. This opens up the entire field of prevention for Alzheimer's disease, because now we're even having clinical trials today where we're testing people that have positive biomarkers like the PET scan, which actually our foundation helped to develop. So you'd screen a population of people in their 50s, find people that have the disease, one of those 47 million people I mentioned earlier, who are not symptomatic, and start to treat them to see if you could actually prevent even the onset of symptoms. And this is really an incredible advance. And yet, as you see on the left, there's just not enough money yet going into diagnostics, which is why we set up the Diagnostics Accelerator. Next slide, please. Now, before I get to the Diagnostics Accelerator, we've been investing in a company called C2N for many years now. They were involved in developing diagnostics, specialty diagnostics for Alzheimer's disease. And just during the last year, they got approval to roll out into the provider community. And this test is available now in 49 states. We're waiting for New York to approve the test. This blood test, a blood test for Alzheimer's disease, is on the market. Doctors can order it. It correlates completely with the brain scan, which correlates completely with autopsy. So we know the validity of the test. It has a high degree of what we call sensitivity and specificity. So imagine, and I never would have believed this could have happened even 5 years ago, but imagine today, a patient can go into the doctor's office, and just like you can get your cholesterol tested, or just like you can get your blood pressure tested, you can go into the office today, and primary care or specialist or neurologist, and you have some memory symptoms, you're really worried, you want to know what's going on, maybe you're thinking about going into a clinical trial, you want to get into prevention, the doctor can order a simple blood test and tell you with, let's say, around 90-95% certainty whether or not you have Alzheimer's disease. That's a revolutionary advance, not just in early diagnosis, but also in clinical trials, because the PET scan that's used to, and I'll get to this in a minute, let me see, the PET scan that's being used now to screen patients for clinical trials, for example, Aduhelm and amyloid, it costs $8,000. And this blood test, and it involves a little bit of radioactivity and takes an hour and a half. The blood test is a typical blood draw. and probably costs around $1,000. So it's going to be revolutionary for clinical trials as well as clinical care. Next slide, please. Now, I mentioned before, we put together, because this blood test is only the first of a revolution in these biomarkers for Alzheimer's. And so the Diagnostics Accelerator, which is one of our AGDF programs, was put together. It's a $50 million effort. Our founder, Leonard Lauder, Bill Gates, Jeff Bezos, Mackenzie Scott, David Dolby, the Schwab Foundation, the Association for Frontotemporal Dementia, and some others have all put in $50 million to develop better blood tests, more blood tests, eye tests, and digital tests for Alzheimer's detection, early detection. And if you go to the next slide, I mean, this thing is amazing. Here's a company called ReadySpec that we invested in that can see the amyloid plaques using rather typical retinal imaging technology at the back of the eye. So imagine the people that are going for routine annual ophthalmology or optometry exams go in and get screened to see if they have Alzheimer's disease, if they want to know. Or if they're having memory symptoms, they can go to the eye doctor and get a retinal test, which would be maybe $150. And we don't know how accurate it is quite yet, but it's one of the technologies that's really exciting. And there's a lot of other uses of this retinal imaging technology that we're looking at. So that's another area of biomarkers besides blood tests that we're looking at. Next slide, please. And then digital technology, which means we're going to use all of the things that, you know, your Apple Watch can do and all these other devices that are out there to look at things like cognitive testing, memory testing on your phone. That's at a research level. We've invested in a company in Australia called Cogstate that's used in clinical trials and is developing an app. And so if you wanted to find out how your cognition was and get a really accurate assessment of it, just like you can get cholesterol or blood pressure, you can go to your app and see how your memory and your other cognitive functions are. And there are going to be other uses of this. Next slide, please. Another interesting digital technology that's being used now is speech biomarkers. And it's really interesting, if you've ever met somebody with very early Alzheimer's disease, you kind of notice that their speech just doesn't sound right. They're using less words. They're constantly kind of struggling for words. Their semantics are not normal. Their flow of conversation isn't necessarily normal. Their pace changes and so on. And so we can use devices like the Apple phone or other kinds of devices and actually look at speech itself on a passive basis or on an active basis. That means we're just monitoring you behind the scenes, or you're actively signed on to some sort of device that looks at your speech. And we can bring this out. Now, this is really important because it's really inexpensive. It can be done daily or weekly or monthly, whatever you like. And it can be used in clinical trials to look at the efficacy of a drug. And it's clinically important. So this is another really critical on the horizon biomarker that we're going to be looking at. So really exciting work. Now, the last thing I want to talk about is this recent, next slide, please, approval of Aduhelm. Now, on the right, I've talked a little bit about the PET scan. And here's images of the PET scan. So on the left-hand column of your screen, it says baseline. And that's at the beginning of this clinical trial that Biogen did in phase 2. And you see the red means positive. It means that when you do the PET scan, the Amyvid PET scan that our foundation helped to develop, and you do the PET scan in patients, you see the amyloid in the brain. You actually see in a living person the pathology. And you can see that all the 4 people in the left column had positive amyloid pathology. On the right, you see what their brain scan looked at after 1 year. And you can see that on the top, the people that got placebo, there was no change or an increase in the amount of amyloid in their brain. But then as people got increasing doses, 3, 6, 10 milligrams, the amyloid disappears. The drug worked amazingly. And all of the drugs in this category of anti-amyloid antibodies actually removed amyloid from the brain. It was effective in doing what it was supposed to do. The real question was, and this was the real scientific question, is if you remove the amyloid from the brain, do people get better? Does their disease slow down? And in this trial, this was one of the first trials. It was really rigorous, what I call modern era trial, where you could actually ask that question because you had the biomarker here, and the drug worked. but did the drug just work on removing the amyloid, or in doing so, did that improve the disease? So it was a landmark trial. And what happened was that in the conduct of the trial, one trial was positive and one trial was hard to interpret. And so the FDA had a really hard time trying to either approve or disapprove the drug. And so they ended up giving the drug what's called a middle ground accelerated approval. That's a regulatory term. That means that the outcome here of removing the amyloid, that the biomarker played a critical role, was reasonably likely to predict clinical benefit. And that's where the controversy starts, because the FDA kind of jumped from, asking the question about, in a real rigorous way, whether if you remove the amyloid, people slow down, to jumping to conclude that it was reasonably likely that they would, giving the drug an accelerated approval, which not only got the drug perhaps on the market, but also required a post-marketing study to prove that the drug worked in slowing clinical progression. And in doing that, if they proved that it did, then the drug could stay on the market. And if it didn't, then the FDA could pull the drug from the market. Now, you've heard, next slide, please. You've heard a lot about the controversy here, and I'll go through these, rather quickly. But the first thing that the FDA asked the committee, that the external committee, and is grappling with, was, are the clinical trial results for the drug actually interpretable? Because as I said earlier, one of the trials was pretty positive, but the other trial was not. And by random chance, you could think of it either way. Maybe the true trial was the negative trial and the positive trial was a random event, or maybe the positive trial was done more carefully and patients had more exposure to the drug. And the negative trial, where there was less exposure, was the one that really wouldn't hold up to proving one way or another whether the drug works. So the first question around this approval is really, are the data interpretable? The FDA's own statisticians, in contrast to the neurologists, the FDA's own statisticians said that the data were not interpretable. Controversy number one. Even if the drug slowed it down in the clinical trial, was that result clinically meaningful? The drug only slowed the rate of progression down over 18 months by about 22 to 25%. And we generally think of the rate of change variable being about 30%, 35%. So it's on the margin of being clinically meaningful, which means even if people take the drug, would it really have a clinically meaningful effect on the patient's disease? Now, some other drugs that are coming down the pipeline, for example, Lilly's drug, donanemab, had a similar mechanism of action, but different, slightly different. And it seems to be more clinically meaningful in the phase 2 results. What about safety? Is the risk to benefit ratio of safety on this drug favorable? Well, 40% of people in the trial had adverse events, and these were detected by MRI, which had to be required in the trial based on previous experience with these kinds of drugs. And these are small, tiny little hemorrhages or evidences of edema, sometimes associated with headache and confusion. And so these side effects are manageable, but they are considered serious side effects, and it occurred in about 40% of people. Was the FDA approval process appropriate? There's some question about how, in this case, the FDA interacted with Biogen in making its decision to approve the drug and recommend approval at the FDA committee and so on. And so that's actually being investigated now, to go back and see what the actual process was, because usually there's supposed to be a real high wall between the FDA and its analysis and the companies. And then finally, even if all of this is overcome, will Medicare and other insurers pay for the drug? And right now they're not. And the cost of the drug, as currently mandated or recommended or paid for, I can't think of the word, the cost of the drug that's approached by Biogen right now is $56,000 a year. And that's not even the total cost of the drug, because you have to pay for infusion centers and so on. So this is a really expensive drug. And we pay for expensive drugs in people that have rare genetic disorders where there's a very small number and there wouldn't be a big societal impact. But here we're talking about a drug that could cost more than $56,000 a year, that could go to 500,000 or 1 million people. And it would really have an impact not only on individuals and their need to pay copays, but also on society, the Medicare drug budget, taxes. If Medicare decides to pay for it, somebody's going to end up paying for it. It'll probably be taxpayers ultimately. So there's a big hurdle here, and a lot of systems like the Veterans Healthcare System, which is the largest healthcare system in the United States, has refused to administer the drug because of its costs. So last thing is, what I get asked is, I have a small practice of patients with Alzheimer's as a geriatrician. What do I tell patients? Well, first of all, we have to find a place in New York City where the drug might be administered. I believe that Mount Sinai is developing that capability here in New York. But my role as a physician is kind of to inform people about the pros and cons of the drug. And then through what we call shared decision-making, the physician and the patient make a shared decision once the patient or their loved one, their caregiver, understands the risks and potential benefits and costs of this drug. And then depending on their decision, I try to enable their wishes. And so I might have some bias in how I present it, but basically I see that as my role, and I think that's the appropriate role. Some doctors are more definitive about it. They have concluded that the drug is not worth it. Healthcare systems have concluded, some of them, that it's not worth it, but others are starting to look at it. So I don't think we're at the final days of this whole thing yet, but it is a breakthrough. The next slide, please. It's a big breakthrough, actually, because Aduhelm is the first disease-modifying drug that's ever been approved for Alzheimer's. It's the first drug that's been approved for Alzheimer's since 2003. It's the first drug that's used in the modern era of clinical trials, all the rigorous methods and biomarkers that we have now. And it's paving the path. The clinical trial program that Biogen put out here and Lilly is putting out really are paving the path for the development of new drugs. As I mentioned earlier, we got inflammation biomarkers and anti-inflammatory drugs going on, and epigenetic biomarkers that are going to drive epigenetic drugs coming to market. And so ultimately, it's going to be, like I said earlier, precision medicine driven by biomarkers with combination therapy. And so the research will go on whether or not Aduhelm is ultimately a blockbuster or not. It's an incremental advance in our clinical research, and I think it's been a very exciting study. And as I mentioned earlier, we have actually none of our clinical trials, except for one program, actually targets amyloid, and the other 34 are all against these novel targets that are coming down the pipeline that are going to address all the other biological, pathological pathways in Alzheimer's disease. And, I've been doing this for 40 years. I've never been more excited about our field and the breakthroughs that we're making now. And I think on the next slide, I'm backed up by somebody you probably all know, who's Bill Gates, one of our partners in our programs. He's been very supportive, and he's happy, so I'm happy. And with that, I'll stop. And thank you very much. And I'm happy to answer any questions that you have.
Yeah, thank you so much, Dr. Fillit. That was fascinating. Let me go to, all right. So anyone who has questions, please, you can type it in the chat box or the Q&A. Let's see. I think everyone's still processing everything you just shared.
There's one
Oh.
Q&A, I think, that popped up there.
All right. Okay, what are considerations for people considering entering a clinical trial versus pursuing existing medications?
Yeah, that's a great question that comes up a lot. Clinical trials can be burdensome, and especially in Alzheimer's disease. Most of the trials require brain imaging, PET imaging, which requires a microdose of radioactivity, not harmful, but some people get concerned about it. MRIs, neuropsych evaluations, which can take hours, repeated visits to the clinic to get the drug. The Aduhelm, for example, is a monthly visit for an infusion. So sometimes if it's not a pill under testing, it can be burdensome to take the infusion. And at the end of the day, people don't know if they're getting a placebo or the drug itself. And if a trial takes 18 months, that's 18 months. Sometimes people look at it, that's 18 months out of the rest of my life, and I'm 75 or 80. That's a big chunk, and I don't even know if I'm getting the drug. So I think it's very dependent on the individual perception of their contribution in sort of an altruistic way about whether or not participating in trials to advance the field is important to them, as well as their conception of the actual potential benefit to themselves. The other alternative is repurposing, where I can prescribe any drug for any indication. Doesn't mean the insurance companies are going to pay for it, but one of the drugs that we've had under we've investigated under funded the clinical trial of called Riluzole, which is an ALS drug, the only drug that even modestly changed the course of ALS, another neurodegenerative illness. And we've repurposed in the clinical trial Riluzole, and I've used it in my practice. And it's all anecdotal, but it's safe. And so if people say, I don't want to enroll in a clinical trial, I don't want to go through that, I'm too frail, I don't want to drive 50 miles to Columbia and then sit there, or whatever it takes, it's hard. Is there anything else you can do for me? So repurposing drugs like Riluzole or some of the diabetes or hypertension drugs, I can't say there's guaranteed there's going to be a benefit. I can only choose safe drugs. And sometimes it's even hard to know if the drug is working. But that's another way that people can at least try something. And I think a lot of families want to try something. At least in that way, people know that they're getting something that has a sound scientific background.
Thank you. The next question, I have been diagnosed with MCI. I'm in my 50s and I feel fine. Should I be concerned? Folks both had cognitive issues.
So MCI in the 50s is actually rather unusual. The average age of onset of dementia, for example, for people with Alzheimer's disease is 75. And usually MCI might start around 70. So first thing I would say is, if you're diagnosed with MCI in your 50s, that's rather unusual. And I would want to know why you have MCI. I would want a definitive diagnosis. So, saying you have MCI is like saying you have anemia. If your blood count was low, you'd not want to know why. You'd want to know if there was a colon cancer or GI bleed from the stomach, or did you have some other kind of anemia? And most often MCI, especially in older patients, is due to early Alzheimer's disease or vascular dementia, or maybe frontotemporal dementia or other causes. So I would recommend, if you've been diagnosed, to make sure that you went through the full gamut of diagnostic tests, like an MRI with volumetrics, maybe a PET scan, an FDG PET scan to look at metabolism in your brain, and even an amyloid test like the Precivity's test that's available now, a blood test to see if this is a case of early, very early Alzheimer's disease, almost in the framework of having a genetic disorder. And I would ask you, if your family members, if your parents also got Alzheimer's disease, or your siblings at the early stage, maybe it's a gene. Because when clinical dementia symptoms or MCI starts in the 50s, we usually look for more rare causes like genetic contributions, and maybe do some apolipoprotein E testing, which we haven't talked about, which can be done routinely as the major genetic risk factor for Alzheimer's disease. And I don't have time to really get into that right now, but I would look at your ApoE genotyping testing, which Quest does, for example, and other major lab companies, to see if that's why you got it early.
Thank you. Any other questions from the group? All right. Well, thank you so much, Dr. Fillit. Have a wonderful day. Thank you for
Thanks for the
joining us.
opportunity. Nice talking to you.
You too.
Bye.
Take care.